Hi, welcome to this video where we're going to be running through the answers to these multiple choice questions that are published by the OCR exam board covering module 2.6, which is about cell division, diversity, and cellular organization. So first step is to find these questions and do them before we review them. So you can either get that from Teams or you can get that from the link below, but you'll have to skip past all the answers to get to the questions and do them first, okay? Right, here we go. Question 1. So we've got this diagram here showing the cell cycle, and it says if there are 40 units of DNA during stage G2, how many units are there going to be in G1? So if it's gone through G2, to get to G1, it's got to go through mitosis, therefore it will have halved. So the answer is B, 20. Okay, moving on. Second one, what about in prophase? Okay, so prophase is in this phase here, right, the beginning of mitosis, right there. And in that phase, the cell hasn't yet divided, so it's still going to be 40. So the answer is C, so therefore that is B. Okay, 3. There are two main processes in the mitotic phase. Which process occurs last? This answer here is cytokinesis. So cytokinesis is the division of the cell, not the nucleus, and that happens at the end of the mitotic phase. Some people sort of say it's separate from mitosis, it kind of goes hand in hand. 4. Here's a chemotherapy drug. Cytarabine is an anti-metabolite, okay, similar to the nucleotide cytosine. So basically what this means is it will be incorporated into the DNA, but then it will kind of stop. It says it stops the making and repairing DNA. So it kind of maybe gets added by a DNA polymerase enzyme, but then it kind of stops the process continuing any further. Sort of working a bit like a competitive inhibitor for an enzyme in a way. So which stage will this be incorporated? Well, it's in DNA reproduction, so that is in S phase. Synthesis, that's C. Okay, 5. Which of these structures involved in cell division is present in animal cells but not higher plant cells? Okay, so this is the centriole. Now, I think actually this needs a bit of explaining because I didn't put it on my mind map. I think I meant to, but I forgot. So you should probably add this to your mind map. So the main difference is, let's make some notes on this real quick. Okay, I'll do it in green. So plant mitosis. How is it different than animal cell mitosis? Well, two things actually, not too much to know. No centrioles, okay? So the spindle fibers in plants are not organized by centrioles. They're organized in a different way. They do the same thing in the end, but there's just no centrioles. The second thing, which you should be aware of, not as frequently tested, but when the cell divides, so animal, when the cell divides, so I'm talking about cytokinesis, in animals, here you have like a cell which already has gone through mitosis. So here's two nuclei in the cell, okay? You shouldn't really shade in biological drawings, but I'm going to. So here's two nuclei here. How does the cell divide? Well, actually, in an animal cell, the first thing that happens is there are fibers of kind of protein filaments that are kind of laid down in the middle. So if this is the cell, what happens is those fibers wrap around the middle of the cell and they constrict like that. So the cell actually pinches in like that until it cuts itself in two, and now you've got two cells, okay? In a plant cell, it's slightly different. So in a plant cell, what happens is you have, it's kind of squarish for a start because it's got a cell wall. So I'll try and draw that like that. We've got those two nuclei. So here's one nucleus over here. Here's one nucleus over here. Now, because it's got a cell wall, it can't really pinch in the middle like that because it's very tough, the cell wall. So what happens is the plant starts to deposit a new cell wall down the middle, and it kind of builds this down the middle. So, yeah, that's basically the difference. Animal cells pinch in and separate, whereas plant cells kind of build a new wall in the center of the cell, okay? Those are the main differences. You might want to add those to your mind map or just make notes of those for sure. Okay, let's move on to question 6. At which stage in mitosis and meiosis do chromosomes always line up at the equator of the spindle? Well, this is metaphase. That's an easy one. D. 7. Spindle fibers play an important role in mitosis and meiosis. Which biological molecules are spindle fibers composed of? Which of these? Okay, well, spindle fibers, hopefully you remember that they are basically microtubules in a sense. Special type of microtubules, and microtubules, you should be aware, are made of a protein called tubulin, and it's a protein. So the answer is B. 8. The diagram shows stages of mitosis in human testes. How many chromosomes are in the cells after meiosis 1 and meiosis 2? Now, this is a kind of a tricky question because it really centers on what is our definition of a chromosome, okay? So it's going to help actually if I just quickly draw a chromosome, okay? So let's just draw in this cell, or next to the cell, what the chromosomes would look like, okay? So in this cell here, we've already gone through DNA replication, so we've got pairs of chromosomes. It's a diploid cell, and the pairs of chromosomes look like this. What happens in first meiosis 1? Well, in meiosis 1, we divide the cell, we separate the pair of homologous chromosomes, or the bivalent, and then in meiosis 2, we kind of get to this stage, okay? So that's what it looks like. Now, we could also, just to recap, let's just remember actually that some crossing over does happen, so it actually might sort of end up looking a bit like this. So we might have had some crossing over between these two here, for example, to generate 4 different gametes. But what does that mean for down here? Well, so after meiosis 1, this is the time when we have a reduced number of chromosomes. There'd be 46 chromosomes here, there'd be 23 chromosomes here. There are 23 chromosomes here, except just each chromosome is no longer comprised of 2 sister chromatids. So this is 2 sister chromatids, and this would be no longer 2 sister chromatids. And if I was going to include that crossing over there, I'd have to change my diagrams up here a bit and include a bit of crossing over there. Yeah. Okay. So the answer is therefore 23 and 23. Is this one? D. Okay, on to 9. Question 9. During which of these stages of cell division do chromatids separate and move to the poles of the spindle? So again, we've got to be really careful about these definitions, chromatids. So when the chromatids separate, that is happening in anaphase of mitosis. But what about in meiosis? When are the chromatids separating? Well, look, we just talked about it up here. It is going to be sort of here in the second division. So it's going to be this one, B. Anaphase of mitosis and anaphase 2 of meiosis. Because in anaphase 1 of meiosis, we're separating the homologous chromosome pair and not the chromatids within one chromosome. Okay? Right. 10. A student examined the cells in garlic root tip and obtained the following data. I'm just getting my calculator ready. What is the percentage of the cells examined in which the chromosomes are visible and consist of a pair of chromatids joined together? All right, visible. So visible in prophase. Yep. And consist of a pair of chromatids joined together. So metaphase. Yep, they're just lined up down the middle. So that's going to be that too. Anaphase, no, they're not joined together anymore. In anaphase, they're moving apart. Same thing for telophase. Interphase, they wouldn't be visible. So we need to take these two numbers and add them together. 74 plus 18 is, hang on a minute, 92. Yep, 92. So, and then we need to divide it by 982. So 92, oops, over 982 equals, can't do that in my head, but it's around about 10%. Excuse me. 92 divided by 982. So it's 0.09. 94. And if I do that as a percentage, times it by 100, that's going to be this one. Okay, so make it a percentage times by 100. Just follow my working. It should be 9.4%. Okay, so that's the one. C. All right. 11. A diploid cell contains 3 pairs of homologous chromosomes. Each pair is heterozygous. Big A, little a, big B, little b, big C, little c, respectively. If this diploid cell, which contains 3 pairs of homologous chromosomes, undergoes meiosis, how many combinations could you produce? So this is all about independent assortment, which is one route that a cell generates, that an organism generates varied gametes. So let's think about this. With only 1 pair of homologous chromosomes, so that would just be sort of big A, little a. How many combinations could you make? Well, you could either make big A, or you could make little a. So you only have 2 combinations. With A and B, with 2 pairs of homologous chromosomes, you could do big A, big B. You could do big A, little b. You could do big B, big A, little b, big A, big B. You could also do little a, big B, and little a, little b. So 4. So it works like this. So with 1 choice, you can do 2. With 2 choices, you can do 2 times 2. And with 3 choices for chromosome, you can do 2 times 2 times 2. Or, if you want to take the maths of this a bit further, where n equals number of chromosomes, so here it's 3, the number of different combinations is 2 to the n. So in human cells, 2 to the 23 is a massive number. In this case, it's 2 to the power 3, which is 8. 2 to the 3 equals 8. Okay, B. 15. Which of the following descriptions of mitosis is or are correct? This is certainly not correct. DNA synthesis occurs at the beginning of prophase. No, it doesn't. It's not in mitosis, it's in S phase. The nuclear envelope breaks down in prophase, yep, and reforms in telophase, yep. The division of centromeres occurs in anaphase. Now, this is a tricky one again because there's these like lots of different words that sound similar. Centromeres, not centrioles. So let's do a chromosome. So here's a chromosome at metaphase, and I'm going to do the centriole in red. Centromere, sorry, I got it wrong there. Centromere is this bit in the middle in red. Now, what happens when it moves into anaphase is that, so this is already attached to spindle fibers. Let's draw the spindle fibers like this, kind of spindle fibers. Like that, so it's attached to spindle fibers, this one in here. And at anaphase, it's pulled apart. For that to happen, we have to duplicate, we have to duplicate the centromere. So it would kind of look like this at anaphase with the centromere there, centromere there. The centromere is still the place where it is joined to the spindle fibers that are now kind of pulling it this way and that way. Okay, so it is 2 and 3 are correct, so therefore the answer is C there. Okay, 13. At which stage of mitosis do chromosomes uncoil and the new nuclear envelope forms? That's a pretty simple one. That's just D, so you just got to know that, telophase. 14a, in which stage of meiosis are chiasmata formed? Well, it's definitely at 1, it's definitely not at 2, so let's cross those out. Definitely not anaphase. Okay. Prophase 1. So yeah, prophase going into metaphase is when the chiasma are happening. Why is the formation of chiasmata is the plural, sorry, chiasmata, an important feature of meiosis? Well, it's all about variation, so which one fits with that? It ensures that the same genetic characteristics appear in daughter cells. No, because meiosis generates varied gametes, and they're not the same as the parent cell. It ensures that the number of genes in the new chromosome changes to produce number of genes changing in the chromosome. No, that's not right. It should keep the same number of genes in the chromosome. It might change the alleles for those genes, though. It provides opportunities for new genotypes to arise. That is the one. Prevents, no, it doesn't, definitely doesn't do that. In fact, it is the thing that pairs them up. So 15C. It causes more variability, more genotypes to arise. 16. Vincristine is another chemotherapy drug used to treat cancer. It prevents spindle formation during mitosis, so what can't occur? DNA replication, that can happen, doesn't need a spindle. Chromosome coiling, that can happen, that doesn't need a spindle. Sister chromatids becoming visible, that can happen, doesn't need a spindle. Sister chromatid separation, that needs a spindle. That's basically anaphase, so that's not going to happen. 16D. Notice that a lot of chemotherapy drugs interfere with the cell cycle because that's cancerous cells divide frequently. So chemotherapy drugs often target this cell cycle because it has more of an effect on cells that are rapidly dividing. It would still those drugs are still toxic to the other cells in the body, which is why chemotherapy drugs have lots of side effects, but they're more toxic to cells that are dividing rapidly, i.e. cancerous cells. 17. All right, we're going to need to do some more maths here. Number of cells, each stage of cell cycle in a root tip meristem. Remember the meristem, in case you can't remember. So here's a root tip, sort of growing down. The meristem is a bunch of cells about here, and those are the cells. They're a bit like plant stem cells, and they're actively dividing, and they're responsible for growth of the root down into the soil. Okay, takes 24 hours to do one complete cell cycle, so how about how long is spent in metaphase? Well, first we need to work out the percentage of cells that are in metaphase. In metaphase, we've got 12 out of 200. So let's do that. So 12 out of 200, that's the fraction of cells that are occurring in metaphase. So that fraction so that's the kind of percentage of cells that are in metaphase. So that means that cells typically spend 12 200ths of the day in metaphase. So now we need to times it by 24 hours to work out how much time they spend in metaphase. So I'll do that. 12 divided by 200 equals, I always do, times 24 equals, so that's 1.44 hours. And if I times that by 60 to get the minutes, that's going to be 86.4 minutes. So that's in hours, times 60 to get into minutes is 86.4, which is B, minutes. 18. How many mitotic divisions of a meristematic cell in a root tip are needed to produce 256 daughter cells? Well, if we start with one cell, we divide it once, it goes to 2. We divide it again, it goes to 4, to 8, to 16. Oh my word, can we do this in my head? 32, 64, 128, 256. There we go, I did it. So how many divisions was that? It's 1, 2, 3, 4, 5, 6, 7, 8. 8 divisions. The answer is A. I think if you've got a scientific calculator, I think you can work that out scientifically in your calculator. You have to do an equation something like this. 2 to the x equals 256, and solve that using your scientific calculator, and then it would give you x equals 8. Okay, 19. What is the longest phase in the cell cycle for human liver cells? Well, pretty much all human cells. Doesn't really matter that they're liver cells. So when you see this question, you might go, oh God, I don't know the answer for liver cells. Just think generally, in human cells, which one's going to be the longest one? Well, prophase is part of mitosis. Interphase is the whole rest of the cycle. Cytokinesis is just a tiny little bit, and telophase is part of the mitosis. You've got to remember that this sort of cell cycle diagram that we're familiar with, whereas kind of mitosis is just up here, then we've got sort of G1, and then we've got S about here, and then we've got G2. So interphase, remember, is all of this bit. Okay, so that's definitely the longest phase. So it's B. Just drawing that. There we go. Okay, 20. Last question. Which of the following descriptions of meiosis is or are correct? Meiosis results in the formation of 4 daughter cells. Correct. In prophase 1 and 2, the chromosomes condense. Yes. Metaphase 1 is the same as metaphase in meiosis. No, because in metaphase 1, what's happening is we are separating the pair of homologous chromosomes, okay? That's what's happening in metaphase 1, and these two things line up and then they separate at anaphase. Whereas metaphase in mitosis, what we've got is we've got just one chromosome lined up down the metaphase plate, and that is being separated, okay? Key difference there. So 3 is not correct, so therefore it's only 1 and 2. So the answer is B. Okay, so that's the end of that. I hope you found those useful. Sorry about the delay for uploading my previous video on the cell cycle mind map. My internet is quite slow today for some reason. Hopefully this will get to you in time, and I'll see you for our next lesson next week, which will be a mind map on, I think, exchange systems in living organisms. Thank you very much, and bye-bye.